So thanks, Ulysses, for inviting me to do this. As Ulysses mentioned, this is a new area for us. And I just like to talk about some of our interests in this area so we can figure out how we can actually address the questions we're interested in and obviously build some collaborations. So this is going to be quite different from things we've been hearing this morning. So, but hopefully this is fun for you as well. So we work on the gut microbiota in my lab. And if you don't know what that is, this is, back down to earth from the stars and every other thing. So gut microbiota are basically the microbes that are inhabiting, humans and also every other creature. And so they, for example, the average person has about 39 trillion bacterial cells in or on their body. And we know that these things are quite important for how healthy people turn out to be, or what diseases they may develop, childhood disorders, older age disorders, and everything in between. And so we actually know some things about how these microbes get into our body. And there's a lot of there's a lot of theory on how this comes about. So for example, in biology, there are these processes we think about. It could be dispersal, as in you get microbes from one environment into the other. So in this case, it will be obtaining microbes from the diet or the environment. Or it could be selection, also called niche selection, where certain microbes are better adapted to the person or animal you're looking at, and therefore you have some microbes which are going to fail to establish or colonize a particular host. And then there are processes such as random drift, where it is not really a deterministic process and things change over time, more or less in a random fashion with a large effect of the environment. And there are also other niche partitioning types of processes where things that are originally there for one purpose can sometimes be adapted to other purposes. So these are some of the basic hypotheses for how microbes actually end up colonizing the host. And generally speaking, why this is important is it has a lot to do with the maturation of the microbial community. So not all microbes are obviously good for us. So we have commensal microbes, which are useful to us, and they're pathogens. And how the final community looks like is determined quite a bit by these priority effects that I talked about in the previous slide. Now, there are some other things that are testable in trying to understand these interactions. So for example, you have on the top here, assembly or microbiota assembly, where you start with a blank slate, essentially, and then you start adding microbial members until you have a mature community. As people or animals themselves become older and they mature. And so you have assembly with the deterministic host selection, where regardless of what order certain species arrive into the host, it is going to be dominating in that environment. And it's going to influence everything that is going to come after it also. And then there are these assembly effects with priority, assembly with priority effects, where the order of the inoculation makes a difference. So you have these two microbes here, for example, which are kind of swapped in their order, and whoever comes first has an upper hand in establishing in the community. And so I actually work on chicken models because in chicken, there are a couple of things that we are interested in. One is it's a major agricultural model, and engineering the microbiota is a big multi-billion-dollar kind of industry because it has a lot to do with how chickens get sick or die and so on and so forth. But it's also a great model to just understand how this process works, because chickens are born sterile because they're an egg and they acquire everything from their environment. So it's a great way, an experimental model to study the establishment of microbial members. So here's some of our one of our studies, early studies, looking at time series data in how the chicken microbiota community is varying over time. So you have different taxon members. So a typical microbiota can have anywhere from a few hundred to several thousand microbes that are colonizing their GI tract. And so these time series data are a great way for us to understand, for example, what was the initial state, and what is the mature state, and what exactly is determining that trajectory. So that's the question we are really interested in, because we want to know what determines the final product in terms of what it ends up looking like. And so one of the advantages of doing work in this area right now is it's easy to generate the kind of data needed for this, because unlike just a decade or 2 decades ago, we can actually generate abundance data for these thousands of microbes in one sequencing experiment, and for obviously hundreds and hundreds of experimental individuals as well. And so here's one of the examples from things that we are looking at. For example, you have this is a linear discriminant analysis trying to figure out which microbes are enriched at the cost of which other microbes. So determining antagonistic relations to the extent possible. So you have things that you might recognize on this list. For example, a common one is lactobacillus, things that you're going to get from kimchi or yogurt or things of that nature. And at the other end, some of these are pretty mundane organisms, but also things such as Alice-Stipes. I want to point that out. It is one of these microbes that are associated with metabolic disease. So it's not a good one, basically. So these types of experiments help us understand how these communities are operating, who are the primary protagonists, and what their interactions are. And what we still don't know is, what is the initial state of these microbiotas, and how do they end up in this place, and how much priority effects are affecting this. And that's where we think some of these, the machine learning and neural network can be very useful to kind of figure out. And so we are trying to predict initial community states and how that affects the final community, and also how the order of inoculation, so the order in which they are acquired, affects the final community. And how does the environment alter these pairwise interactions, for example? And this is, as I said, something that could be very useful for human and animal health through microbiome engineering, as they call it. So we have done some initial work on it. But this is like, something we started only recently. So hopefully we'll have more to talk about next year. One of the things we've been doing is, are pairwise interactions stable if priority effects are in action? And trying to estimate, get the parameter estimates for birth and death rates for putative antagonistic interactions, and which taxon pairs are predictors of the mature community. So I also realize that most of this may be completely new to some of you. So please stop me, or I'm happy to stay back later and discuss more of this. So I know this is quite different. So commonly, these types of questions are, addressed with the Lotka-Volterra equation or model, which is a common first order ODE. And it describes these interactions between, as a predator-prey interaction, and only with these 4 constants: birth and death rates of predators and prey, alpha, beta, delta, gamma. And for microbes, this is an okay approximation, because it's not perfect, because there are thousands of species interacting in this system. But we can kind of use this to understand some basic interactions here. So because we can have time series data, we can get an idea of abundance of microbes over time, and we can try to estimate some of these parameters, hopefully with this type of data. Now, in this case, as I mentioned, there are really no prey and predator like strict interactions, because microbial interactions can come in a variety of flavors. They could be competitive, they could be mutualistic, commensals, parasitic, or predation. So there's a whole range of interactions possible here. And the regular Lotka-Volterra model is not really a good approach, but in this case, that's the the thing that we've worked with so far. And we did this, some of our initial work with using the differential equations package in Julia to fit our data to the Lotka-Volterra equation. And then we use the Turing package to use, Bayesian parameter estimation from MCMC. So here's some of our data that we use to do this work. So you have these two species I kind of mentioned earlier, Lactobacillus and Allostypes. And in these two experiments, we have all, basically the same species we're looking at, but with little differences. In the first instance, we have these two species which are arriving at the same time. And you see that how this interaction plays out over time. In the second example, we have one of the species arriving earlier because this other one was not introduced at the very early time point, and Allostypes in this case takes over this community and retains their advantage over time. Whereas in the other cases, it's quite the opposite. So these data are saying that priority effects are clearly important. It does matter. So what we are interested in, what exactly changes in terms of their birth and death rate. So, so when we ran this with our the differential equation package, so here's what we kind of found. So we were able to get some of these parameter estimates here. So you can see here species A and species B, which refer to Lactobacillus and the Allostypes here. So in the first condition, where both of them are arriving at the same time, you have a higher birth rate than mortality for Lactobacillus and nearly the inverse for Allostypes. So it seems like the Lactobacillus is able to maintain a competitive advantage when both of them arrive at the same time. And when both of them are not coming at the same time, so Allostypes comes in early, then you have, in this case, pretty much a mirror image of that result going on here. See they now have a higher birth rate, lower mortality rate, and a higher mortality rate for Lactobacillus in this case. So that is one of the things we are observing here, contributing to this, difference where Allostypes maintains this competitive advantage. Now, obviously, this is very, very basic stuff that we've kind of gotten into, but even this is new in this field because it is not something people have been able to estimate or look at. And why does this matter? So why it does matter is the order of inoculation ends up altering the function of the mature communities. So besides, generating significant differences in the alpha and beta diversity of the communities themselves. So the membership and the function of these communities end up being different. So, and by that I mean the other 500 to 1,000 species that we see there, they end up being, completely, not completely, but about 30% different, which is quite a bit if you know microbiota studies. So where does that leave us? So basically, one of the issues or the main issues we would like to work on next is we don't know which pairs are truly antagonistic. And I think that's probably not the best way to define these interactions because there are all these other possible interactions. So we need a generalized linear Lotka-Volterra approach that can consider multiple species and multiple interactions among them. There are some packages in R, for example, right now, which can model these interactions, but they are not very good at estimating the, for example, the initial states or kind of discovering some novel insights from how exactly they're ending up at these final states or predicting what the final state might look like. So I think that's one of the areas where the integration of this training approach and neural networks could be quite useful and something I'd love to hear from people who know more about this than I do. And we also don't know how important certain pairwise interactions are. So we've arbitrarily picked two of these interacting species. We actually don't know which ones are the most important one. And this goes to the concept of something called keystone taxa in Microbiota Assembly, which once they arrive there, they have a dominating effect on whatever happens next. And actually, I think that is the most critical and exciting area that needs to be done, research that needs to be done. If you can figure out keystone taxa from initial states and existing data sets on what the final state looks like, that is going to be an amazing advance in this area. And it has like amazing applications for, again, diagnosing human disorders or animal disorders, outbreaks in natural systems. This is like a very broadly applicable question. And so that's pretty much where we are. And I'm happy to discuss this in more detail and would love to have students or postdoc and obviously the faculty in the SciML group who would be interested in working more on this problem. And happy to answer your questions.
Thank you, Giri. I was muted, sorry. So this was a great talk. Of course, I wasn't aware of your work on microbiome, the microbiota, and I think this is great potential, and obviously how you could use neural networks to let's say start from data and do the inverse mapping, right? So can you discover hidden quantities of interest by using the data plus, let's say, the predator-prey model or any other kind of model? Right now, of course, people are just using traditional machine learning. People are just using, data-driven to do this. So, I mean, obviously, you don't necessarily want to use the neural network just to solve the equations. You have good solvers to do that. But the ability of the inverse problem, solving the inverse problem is very promising, I think. And so, any questions from the audience? We have plenty of time. This is a good change of pace, because we were talking only about engineering and science, but biology is a big unexplored territory here for SciML, I think.
Hi, this is Eduardo. I have a quick question.
Yes.
Eduardo here in Petroleum Engineering. It seems like the very last bullet or the last bullets of your conclusions, then you need I have a question on, are you looking for just feature selection processes? Do you actually need to go all the way to neural net to discover the independencies of those features or the pairwise features that you call? Or can you just rank those features in such a way that which are important to contribute to your model? Right.
Yeah. So actually, that's an interesting question. So actually, I'm not even entirely sure that. I mean, ranking the pairs in importance would be good, but the key issue here is, and the community who work on this, they are focused on these hub species or keystone taxa. So it may not even be a pair, it could be just one species. And that's the thing we don't know. And it's not, again, we started with Lotka Volterra because that's kind of an easy starting point, but I'm not even sure that's the best approach.
Okay, very good. Thanks.
Yeah, I guess the question was the model reduction, right?
Yeah, I mean, going into that lines, yes. That's not only model reduction, but the feature selection process. It seems like you have lots of properties, right? Pairwise properties that one can correlate to each other.
Right. Yeah. Got it. Yeah.
That's right. I mean, do they know, Giri, the differential equations that govern this? So you're using the prey-predator model.
Yeah.
Are there other differential equations people use?
I've seen some people using the delayed differential equations as one of the potential approach, because you have, especially for this case, I don't know if that is even answering your question, but that's one of the things I've seen in the literature recently, saying that could be valuable because you're looking at a prior state and how that affects some from future states.
It's a time evolution problem, exactly.
Yeah, yeah.
So that there should be some form of knowledge of this process, this time evolution. Have this species, and yeah. So this is an interesting problem. You know, time evolution problems are very nice. Any other questions from the audience? Do we have a lot of people, Giri, do you think, in the College of Agriculture interested in these problems?
In this particular problem?
I mean, applying scientific machine learning.
I think so. I mean, a lot of them work on these, dynamic systems. Perhaps they are not familiar with what's possible, and I would think so. I mean, I know. I mean, we saw, gene regulatory networks is something I myself in another, domain work on those types of questions myself. I know there's a lot of other people who would find that valuable.
No, James Sky is not in agriculture. He's in vet.
Yeah, he's in the vet school.
But he also showed interest, and yeah. So I guess there's plenty of potential across the tracks to do this, and not stay only on this side of the track.
Definitely, I think in many cases they may. I mean, again, even I guess I'm an amateur at this, but this is a steep learning curve for even me. I have a little bit of a computing background, but if you imagine somebody who's not used to thinking of this, I think that they're not going to.
That's why collaboration is
possible.
That's why collaboration is really important. Yeah. That's right.
But yeah, I think there's a lot of other very interesting problems that you could bring to. Use.
What you already did is very impressive, using the Julia framework. And of course, we are a little bit of competitors. We use TensorFlow, not Julia. Maybe we can interest you in TensorFlow. I mean, I have no prior motives or for using TensorFlow, and I do not collaborate with Google or have any stocks in Google or anything like that. It's just because we started using TensorFlow, and now we have built our framework on TensorFlow, which is TensorDiffQ, and it's an open package like Julia, and so